and then share. You can share you can see the screen, right? Okay. So we are discussing this topic, research methodology. Now in this what happens when we are discussing about, let us say, bioinformatics research. Okay, bioinformatics research. Okay, so it could be bioinformatics research, it could be, let us say, bio simulation research. Okay, that is a better terminology. Okay, this is one kind of research, and one more kind of research is, let us say, wet lab research. Okay, so for both the research types we do need something which are called raw materials, or let us say shall I say materials and methods. These are needed, materials and method. Okay, for both type of research. Okay, so what happens, we are having this particular thing, we are discussing research methodology, where we will be discussing two types of research methodology. One is the wet lab based research, and another is, or this is one biosimulation research, and the other one is wet lab research. Okay, for both these two types of research we do need something which are called let us say, we will call it our materials and methods. This is needed for both kinds of research. Okay, so these are needed for both kind of research. Okay, so these are needed, these are the interconnections, how they are interconnected. Okay, so again we are for research methodology, there could be, in biological sciences there could be two different types of research, one wet lab one, and another is the biosimulation research. Now how these two are different, that is the $1,000,000 question, and which one you should select. Okay, so what happens, the whole thing depends on the mainly on the materials and methods section. Okay, so let us see what are the materials that are needed for biosimulation research. Okay, so first and foremost we will talk about the materials for this, and then we will talk about materials for this. Okay, so one by one, for biosimulation research you need to have a first and foremost the hardware. If we are talking about, we need a something called workstation. Now what is a workstation? A workstation is a high-end computer which is nothing but high config computer. Okay, which will be connected to a high speed network. Okay, it should have sufficient storage, high RAM and ROM. Okay, and then we should have one more thing which are the biological database. Okay, then we need the third one, that is, these are, we are talking about the materials. Okay, the third one which is needed are tools. Okay, so these tools are basically two types. Again, these tools are either shall I say server based grid computers. Okay, for example, example. We will give an example now. For the example part, we will go to this. We will go to this. This is a server based what do you call grid computer. Okay, so this is the example, so we'll put it as an example. This is an example of grid based or server based grid computer. Okay, so that could be an example. So other tools can be downloadable softwares. Okay, so other tools could be downloadable software. Okay, so what we'll do is we'll put a different color to it. Let us say red. So this could be your downloadable software. Okay, so we'll give an example. Let us say examples like Schrodinger. Okay, then we can have examples like Rasmol, etc. Okay, so these are the mainly tools and this thing materials. Okay, so we'll put a bit color here, materials, and then we'll come back to methods. So these are again materials. So here what are the materials we need? Okay, for biological these things, first and foremost we will need reagents. Okay, then we will need kits, various kinds of kits. Then we will need samples. Then we will need many a times, not always, live cells or tissue. Okay, then we will need 5. We will write it like this: massive infrastructure is needed. Okay, why massive? Because equipments, no, which we have not spoken of, these equipments could be as large as very large. Okay, so why we have said massive infrastructure? I'll give you some examples of massive infrastructure. Okay, and you'll understand what is this thing of massive infrastructure. Okay, so massive infrastructure could be put a little smaller, otherwise no, I'll not able to complete. So why we have said massive infrastructure? We need like biological safety cabinets or safety cabinets, which will be very, very large. Okay, then we may need high-end facilities like electron microscope. Okay, so we'll write it like this: SEM or TEM. Means there are two types of microscopes: scanning electron microscope and transmission electron microscope. So which needs a whole room kind of thing. Then there could be like n number of other large equipments. These are all needed. Okay, in wet lab research. Okay, so what is the comparison? The comparison is in wet lab research you need to have multiple different things which should be there. Okay, so if we recapitulate a bit, so what we are talking about? We are talking about the research methodology. So this research methodology has two different types of research: biosimulation research and wet lab research. So both of these kind of research need some materials and methods. Okay, so both biosimulation research and wet lab research both. So we'll see how these two things are different. So when we talk about the biosimulation research materials, then we see that we need hardwares like workstations, and then high configuration computer, high speed network, high RAM and ROM. Then we need biological databases. Then we need multiple tools, which could be either server based grid computers, or it could be downloadable software. Okay. So both the server based computers as well as downloadable softwares you are aware of. Now in materials for wet lab research could be multiple: reagents, kits, samples, live cells, tissues, and massive infrastructure is needed. Okay. So when we talk about massive infrastructure, it means that there is huge need of investments. Okay. Now let us see what are the reagents needed. So these are also very highly costly. Okay. For example, if consider that you will be doing something called identification of protein in a localized tissue, then you must need to have fluorescent tagged antibodies which will be used to identify specific type of cells. Okay. Which are nothing but also monoclonal antibodies, very costly. Okay. Then you may need what do you call radio labeled probes. Okay, depending on the work you need to do, radio labeled probes. Okay, then you need to have some specific proteins such as recombinant shall I say growth factors, etc. Okay. So these are all very highly costly things. Now which kind of kits could be there? So kits could be like, okay, so when we talk about kits, it will be like kits like let us say DNA isolation kit, then Western blotting kit. All these things are needed. Okay, and number of other things are also there. Then when we talk about samples, what are the examples of samples? This could be biological samples. Okay, so we'll write it here itself. So biological, oh no, we have to come here. So biological samples like it could be urine sample, then blood sample, then could be sputum. Okay, sputum means some kind of body fluid, or there could be CSF, cerebrospinal fluid. Okay, and n number of different things could be there, isn't it, which are very difficult to collect. And you also need to, here is very important thing, you need to stick to ethical guidelines as well. Okay, there are some ethical guidelines before collecting all these things. Then there could be live tissue and cells, which can again be biopsy samples, Then there could be for example viruses, so on and so forth, isn't it? So this makes the whole exercise very difficult, and there is a need of huge what do you call, a large group is needed to bring about a successful wet lab research. Okay, so we'll give you an example of a successful wet lab research. In order to do so, what we'll do is we'll go to NCBI. So this thing everyone knows. Okay, listen carefully how you need to identify a biological research. Okay, so this is our NCBI database, everyone knows. Is there anybody who doesn't know about NCBI? Everyone knows, isn't it? So we are having a database, a global database, where you will find uh, n number of things: proteins, nucleotides, genes, genomes, then what else? N number of things are there in this NLM database. So what we'll do is, in order to see an example of a wet lab research, we'll change it to something called PubMed. Okay, then here we'll write, okay, and okay, so we'll search this name and we'll figure out a wet lab research, an example of a wet lab research. Let us hope that the thing will work out. Many a times, because the load is very high on this database, sometimes it will respond slow, but most of the time it will respond quickly. We're still showing protein, but we have converted it to okay. Yeah, that's a better idea. Okay, so I'll give you an example of let's see whether it can. Yes. Okay, so this is an example of a research which is completely wet lab based research. Okay, there is no simulation at all. Okay, in this, as you can see here, preliminary screening for in vitro anti-enteric properties of a traditional herb, Dillenia pantagina fruit extract. Okay, so we conducted this research long back, approximately in 2014. So it has been like more than 10 years. So what exactly we are trying to figure out? We are trying to figure out different fractions of a fruit, and we will study the antimicrobial susceptibility against a specific microbial, what do you call, fraction. Okay, not all, because what we have mentioned here, we are trying to figure out what is the anti-enteric properties. So when we talk about anti-enteric properties, we are essentially talking about what? We are talking about the disease of the gut or the what do you call, alimentary canal. So many a times what happens, there are two major bacteria or pathogen which causes what do you call, enteric discomfort in tropical area like India, Bangladesh, Nepal, Bhutan. Leave it Nepal, Bhutan, then Pakistan and this area. So the main two organisms are E. coli is one, and another is Helicobacter pylori. So Helicobacter pylori is a bacteria which causes high acid efflux in the stomach, which eventually leads to stomach ulcer. I am audible to everyone? I am audible, right? Everybody is there? Okay. So what happens? We want to find out whether a plant extract is effective against these two bacteria. One, E. coli, coliform bacteria. Another is Helicobacter pylori. Okay, very this thing. Acha. Now, in order to do so, what we need to have? We need to have a assay area which is very sterile, completely sterile, which is also known as this biosafety cabinet. Okay, can you remember biosafety cabinet we have spoken about? This is the biosafety cabinet thing, very important one. Very costly also. What does it mean? It means that there will be an area which is completely sterile. Many of you might have seen in your lab small area where ultraviolet ray is also there, then this high efficiency particulate filter is also there, and it gives an area where you can do microbial this thing inoculation and all. Many a times you can have a very large unit or large cabinet where a large area is very sterile, completely sterile. So in that, what we tried to figure out, we tried to figure out that the plant extract which has been isolated in 2-3 type of abstract. For example, here you see DP47 was separated by 30% ethyl acetate. Okay. Then one more this thing is there. One is isolated by 10% methanol. Okay. Then there is, I think, 3 fractions: 47, 43, DP47, DP43, DP50. Okay, and one more was DP50. Yeah, DP50 was isolated by butanol. So what happens? All these solvents, no, these are non-polar solvent. Here you see ethyl acetate is a non-polar solvent. Okay, the ethyl acetate in chloroform. Okay, it's a what non-polar solvent. Then this, again methanol is a non-polar solvent. Methanol and dimethyl sulfoxide both. Then there is methanol alone and there is butanol alone. These are alcohols basically. So we isolated the plant extract in these 3 this thing what do you call inorganic solvents, and then we found out if there is some amount of what do you call antimicrobial activity. So for that we needed some what do you call reagents, kits. For identification we need to have fluorescent microscope, then multiple different things were there. Okay. So this gives you an idea that how what do you call this what do you call wet lab will be looking like. So it will take a lot of time. I think I have mentioned how much time we needed to conclude the whole study. We have not mentioned in the full study, it was been mentioned. Okay, so if we want to go to the full text, we will go to the full text. Here is the journal and the full text. Some images are also there, you can see this. Some images were there. Some images were also there, so right now these are not visible. So what happens? This kind of study needs a lot of time. Okay, lot of time, lot of money also. You need to have external research funding. Like this particular study needed at least 5 lakh rupees of funding, equipments, reagents, and see how many people. 1, 2, 3, and a group of my student, like your group. You yourself is a group. Similarly, Shurajit Debnath, group 1 students. So many students were also there. So everybody's name was not able to, we could include. Okay, those who have contributed significantly, 1 student and another faculty, these names were there. So this is how a wet lab study is being seen. Okay, so now we can see how a completely simulation study will be seen. Okay, so we'll give you one more example. So here we'll give another one which is completely simulation study. See here. Okay, here only two faculties names are there, one this and another this. So and the study was very massive. See, many figures are also there. It was also a massive study. All figures, 7 figures are there. So it was also a massive study, but despite of the massiveness of the study, it was being handled by 2 persons only. Okay, that also the whole study was conducted without any funding at all. No funding was needed. Okay, free of cost, you can say. Free of cost in the sense, effort was there, computational resources were being used, but not research. These things were not there. This whole wet lab thing was not there. Okay, high-end equipment and all such things were not there. So this is how, what you can do is you can eventually start. What was the topic? Structural basis of heterogeneous phenotype of ERG11 dependent azole resistance in Candida albicans. So what happens? Candida albicans is a fungi. Okay, it's a dangerous fungi. It causes multiple different diseases in human being. First and foremost is candidiasis, vaginal candidiasis in females. Okay, it's a dangerous condition, dangerous, and the person will definitely die. So what happens? These fungi was already been treated by drugs or antibiotics which are known as azole. Okay, for example, clotrimazole. Clotrimazole is again a what you call azole drug. Okay, but what happened later on? It was been found that this Candida albicans, pathogenic fungi, has been become resistant to this particular antibiotic, and therefore new antibiotic you should start developing. Okay, and what is the problem? The problem is once one antibiotic is becoming resistant and you have to design a new antibiotic, you need to invest a lot of money for this. Okay, it needs a lot of money to be deployed for production of a new antibiotic, and then whole lot of study, phase 1 clinical trial, phase 2 clinical trial, phase 3 clinical trial, and all such data should be maintained. It needs a lot of money, and eventually what happened? The cost of the antibiotic increases. Understood? So if a single antibiotic is useful, what would happen? It will be very less costly. But once then antibiotic becomes resistant, then you need to develop a completely new antibiotic, which causes a lot of problem. So in this study, I tried to identify how this azole resistance develops. Okay, so whole lot of study were there, and multiple different things were been given. So here you see some figures. Okay, figure 1. Okay, we'll go to the full research paper. So we'll go to Springer. So the other study was been published by Elsevier, which is a global manuscript or science publisher, top level science publisher. This is again a top level science publisher which is called Springer. So here you see, this is the background and all. We'll come back to the images. Here you see one full size table, you can see. So in this table we identified what are the different amino acids present in the whole protein, ERG11 protein. Okay, then I want to show you some images. Okay, here is the this thing of what happened. There is a method called molecular dynamic simulation. In that, what you do, you have designed a protein, right? By Rasmol, you have seen a designed protein. So if you shake this protein in vacuum, many parts will start moving very rapidly. It gives you an idea which part is a active site of the protein. Okay, usually if there are some rapid movement in some part, it indicates that these are structurally important regions. Okay, this is one analysis, this is one more analysis. These are the important analysis. This is the structure of the whole protein, where ERG11, the enzyme which is responsible for, I'll give you the context. This azole resistance, no, that means resistant to azole drug is due to a mutation in the ERG11 protein. Okay, now ERG11 protein is responsible for production of cholesterol inside the fungi. That particular fungi cholesterol is called lenosterol. In human beings, cholesterol is known as cholesterol. In fungi, cholesterol is known as lenosterol. It's a sterol molecule only, but a bit different from the cholesterol. So we studied the enzyme first, and then eventually we started to find out what is the this is the functional amino acids. For example, tyrosine 64, lysine, leucine, phenylalanine, serine, histidine, proline, leucine, phenylalanine, tyrosine, histidine. These are the amino acids which are present on the active site of the protein. Then there is also a prosthetic group. Yesterday we have spoken about prosthetic group, remember? So this is the prosthetic group, heme prosthetic group. In this enzyme ERG11, heme prosthetic group is also there. ERG11 is the name of the protein. ERG11. This is a metalloprotein. Okay, where a heme prosthetic group is there, and these are the active site amino acids. Okay, and then this is the whole structure of the protein. There are two access channel, as you can see, access channel 2, access channel 1. Through this channel only the drug enters, okay, or the substrate enters, Which drug? Azole drug. This azole drug we are talking about, no? This azole drug enters through this channel and then inhibits it usually. But due to the increase in the diameter of the channel, what happened? The drug enters through access channel 1 and comes out by access channel 2. And therefore, the bacteria is not killed because the enzyme is also not get inhibited, because previously the channel was very small, but due to the what you call mutation, the channel has become very wide, and therefore the drug is there. The drug will enter through access channel 1 and by access channel 2 it will come out. So beautifully explained. Okay, understood. So this is how your what you call, and then we did this thing also, phylogenetic analysis of the protein, how it has been produced, how the evolution of the particular protein has been occurred over the time in this fungi, Candida albicans. That also we explained. Multiple different analysis we did, and eventually it was been published also. Okay, in a very reputed this thing. So why these are the two contrasting research papers? One completely biological, there is no analysis, no simulation, and it is completely simulation based. Okay, so this is how we can make you understand that how biosimulation research work, which needs some substantial amount of time, but the requirement for infrastructure is much less. Okay, on the contrary, wet lab research requires a lot of reagents, kits, live cells, tissue, and massive infrastructure as well. Okay, so this is the thing that I wanted to tell you. Okay, so what happens?